Hello, everyone. Welcome. It is just past 11, so we want to get started. I am Dr. Eaton, Dr. Greg Eaton in the Biological Sciences Department here at Rowan, and my co-host is going to be Dr. Janine DeStefano. She'll also be here, and yeah, we want to welcome you to this installment of the seminar series, and we're really excited to hear from Dr. Sarah Wright. Before we jump right into the seminar, I do want to point out we have two more seminars left in the Department of Biological Sciences seminar series. Next week, that's 2 weeks from now, Dr. Christine Blavy-Haas is going to be coming and talking about zinc chaperone for metalloprotein synthesis. Or biosynthesis. I'm really looking forward to that one. She is from the Brookhaven National Laboratory, and I know that they do summer research internships, and there may be some possibilities for some of our students to head up there. So that I'm looking forward to that seminar, and I'm looking forward to hopefully some things that may come out of that. And then we're going to finish up with our final career seminar of the semester with Kendra Flores, who is a genetic counselor. A number of us are graduating or will be graduating shortly in the next year or two, and not everyone is going to become a PhD researcher. So there is other things you can do within biology. And part of this seminar series is to learn about cool research, but to learn about really cool opportunities, careers that you can do with your bio degree. So we hope that you'll join us and learn about what genetic counselors do, and whether it's something that seems interesting to you. So I'm going to stop sharing my screen at this point. I also want to point out that we have the Rowan Bio Events calendar on Google Calendars. If you use Google Calendars, you can click on that or follow the QR code to go to it, and you can add that to your calendar so that as long as you have the button checked, you can see any events that are somewhat interesting to bio majors at Rowan that are coming up. And hopefully you'll find that useful. Otherwise, let's move forward. So Dr. DeStefano is going to be collecting all of your questions throughout today's talk. So please, if you have any questions for Dr. Wright, whether that's about her research or her career journey, please send that directly to Dr. DeStefano. She will collate them and we'll ask them towards the end of the talk. So the way this talk is set up, Dr. Wright will be telling us a little bit about her career journey and what led her to become a scientist in the first place. And then she'll also obviously spend some time telling us about her research. So for those of you who don't know, Dr. Wright is a lecturer here in the Biological Sciences Department. She's been here since 2019. Before being at Rowan University, she got her PhD in evolution, ecology, and population biology from Washington University in St. Louis. However, before that, she got her bachelor's in biology. She always knew she loved biology, as I'm sure you'll find out shortly, but she also always knew she loved teaching. She loved interacting with students. Even as an undergraduate, she got her high school teaching certificate. So I think that those of you who've had her in classes can probably attest to the fact that she really enjoys teaching and has a passion for her students. And I think that the rest of us will be able to see that come through today. Another thing really importantly that I want to tell you about Dr. Sarah Wright is last year she got the President's Award for Excellence in Innovative Instructional Delivery. So what I mean by that is, if you haven't had her, try to take a class with Dr. Sarah Wright. You'll definitely be surprised and happy with that. So I will stop talking and turn it over to you, Dr. Wright.
Thank you. I hope I can live up to all of that. And I did see some names that look familiar. So if you are one of my former students, hello. It's good to see your name, even if I can't see your face. It's a brave new world with all this Zoom stuff. All right. So I think my title slide is showing, and I think you can hear me. Can I get just a visual thumbs up from folks telling me everything looks good? Maybe? Yes? No? Am I there? Can you hear me? Oh, okay. I was like, I'm not seeing anything. All right, thank you. So I feel like I've given this same talk in different versions, and every time I give it, the title changes a little bit. So right now it's combining genetic sequencing and field experiments to study white clover adaptation in diverse climates. But I think what I really want you to get out of this talk is that my research and my interests are broad and integrative. And so I'm not just studying evolution or just studying ecology or just studying plants, but I'm studying how plants evolve and the genetic basis of that process and which alleles are favored in different environments and stuff like that. So I always have trouble coming up with a title, but hopefully it will become more clear as I explain more. I do have a little outline for this talk. So I'm going to tell you a little bit about myself at the beginning. I will then talk about my PhD research. After that, we can do a Q&A. And so please feel free to ask questions about just my life or my research, whatever. If I say anything that sounds interesting, feel free to ask. Those should go in the chat box. I think they should go to Dr. DeStefano if she's there, although I'm sure Dr. Eaton would take them if she's not there yet. And then at the end, after the Q&A, if we have time, I do have some slides and some pictures of extra things I did in grad school that are honestly just kind of fun. And there's a lot that you can do in grad school that you might not know about. So I'd like to share that if there's time. All right. So a little bit about me to start. And I have this kind of disclaimer slide. I get told I look young a lot, and maybe I'm flattering myself, but I am older than you might think. So I like to remind students that the work that I'm presenting took basically a decade. I'm going to start as an undergrad and keep going through PhD and beyond. So research takes time, and this doesn't just happen overnight, even if I might look young. Again, I might be flattering myself. When I started college, I also had no idea that I would go to grad school for a PhD. Most of my family is in business. One of my brothers did math and computer science, but nobody really did research. My grandfather had a PhD, but he had passed away when I was like 1. So it was new for me. And I learned about grad school and I learned about what you could do in research while I was an undergrad. I definitely would not have predicted the past decade of my life when I was your age. So I just want to encourage students to stay really open-minded to possibilities and opportunities that are going to pop up unexpectedly. I've lived in 3 different states. I grew up in Texas, but I've lived in Missouri and I now live in New Jersey. And I would have never guessed that, but it's been delightful. And I've also studied abroad in Costa Rica, and I also would not have expected to do that. And so stay open. Also, at no point have I had all the answers, and I still don't. If people ask you, what do you want to do? What do you want your career to be? It's okay if you're not totally sure yet. I knew I liked teaching. I knew I liked science. I didn't know how that was all going to play out. And I also don't know how it will play out in the future. So I encourage students to talk to older adults, talk to people who have jobs that sound cool to you, ask lots of questions. You're already at this seminar, so that's good. I went to so many seminars as an undergrad, and I learned so much just about what was possible from those seminars. You'll learn a lot about what you like. You'll also learn a lot about what you don't like, and that's almost equally important when you're trying to decide what to do next. So I try to learn something from every experience, and that's true for even the bad ones. I like to talk about Costa Rica, and I have all these great pictures, and it was a great experience, but it was really hard for me too. And I also learned there were a lot of things I didn't like about field research. But I learned a lot and I met a lot of friends. Last thing I'll say is that I'm also just a huge advocate of counseling and therapy. I think it's important to be open with that, especially these days. I think I'm very happy with where I am in my life now, but I've certainly had ups and downs. And I like to talk about how plants are affected by different environments, but I also have learned a lot about how I am affected by different environments, and counseling and therapy helped me a lot to better understand myself and better understand why I feel and respond the way that I do in different settings. So life is a journey, and you're at a really exciting stage if you're a student and even if you're not. All right, so I grew up in rural East Texas. So I grew up east of Dallas. I've got it circled on a map in a little place called Holly Lake Ranch. I grew up on this little, if I move my cursor, can y'all see that? I think so. Okay, I grew up on this little lake, and my very first science experiment was testing bacteria levels in that lake. And this was my high school. It's surrounded by forest across the street from a cattle farm. There were 84 people in my graduating class, and because it was so small, I could kind of do everything. And so I was a cheerleader on Friday nights, and then I did math and science competitions on Saturday morning. And then I was really active in the FFA, where I raised meat rabbits, but also I fell in love with plants, and I started doing floral design and floriculture, plant judging. And that was really where I just started to gain an interest both in math and science and in plants in particular. I decided to go to the University of Texas at Austin. It's a big, awesome school. I wanted to go there. I didn't really think that hard. I didn't look at that many colleges, if I'm honest. I was from Texas. I loved the school. My parents went there, and that's where I went to. And so while I was there, I knew I liked math and science. I got my Bachelor of Science in biology, but as Dr. Eaton said, I also got teaching certificates in math and science. And so people would ask me, what do you want to do? And I just kept saying, I don't know, research or teaching or both, I'm not sure. So I did both for a long time. One of the good things that I did that was unexpected that got me into research, and honestly was strongly encouraged by my mother. My mom really encouraged me to apply to this honors program. And through this honors program, I got to do research all the way through my undergrad. I encourage students, if you're interested in research, to try to get involved early, start making contacts early, even if you're just talking to professors about your interests. And the more experiences you have, the more you'll learn, and the more you'll be able to talk about those in later interviews and things like that. So I was lucky to get started really early. And as a freshman, well, focus on the one on the left. I meant for the phage to pop up first. As a freshman, I studied this T7 bacteriophage, and in particular, I would adapt it to different types of E. coli cells in a lab. And then I sequenced some genomes, and I looked at some of the mutations that popped up as these viruses adapted over time. I thought that was kind of cool. What I really liked about it was that I was studying evolutionary genetics. I was looking at the genetic changes that were occurring as these organisms adapted to their environments. And I thought that was awesome. But the reality of my day-to-day was that I was in a lab looking at cloudy liquids, and then clear liquids, and then cloudy liquids again. And that just kind of became boring to me. I missed plants. I missed being able to see the organisms that I was really studying. So I switched labs, and I started working in a plant lab, and I worked with this plant genetic model organism, Arabidopsis. But I didn't just work with the clone version that everybody grows in their labs. It's like Columbia, if you know anything about, that's like the wild type version. I focused on wild accessions. This is a plant that grows all over Europe and Asia. And I was fascinated by all of the variations that I could see. I could see the plant I was studying, and I wanted to know how all of this variation was maintained and which variation could be useful for adaptation. And so this sort of gets me started in my research journey, because as I was thinking about natural variation and adapting to diverse climates, I started reflecting a lot on how there are a lot of species that occupy really broad geographic ranges. Wild type doesn't really have much meaning in some cases. These species have populations that can experience really different selective pressures. And so they're not all the same. You have different varieties in different areas. Selection can differ in abiotic factors. So here we're looking at temperature, or we could think about rainfall, but there are also biotic factors, biotic variables that vary across the environment and that can impose selection as well. And so these can be things like for plants, organisms that are maybe eating them or pollinating them, helping them to reproduce. And so it's really all of these factors together that impose selection on organisms and that can, in some cases, lead to local adaptation. So I studied local adaptation sort of in every project that I did. I just looked at different systems or asked slightly different questions. Local adaptation is when divergent selection, so selection in opposite directions, causes populations to evolve a higher fitness specifically under their local environment or their local habitat. This produces what we call a population by environment interaction. And whenever I see the word interaction, I think of an X. And so you can kind of see that X on the screen. We're looking at a graphical or a figure representation of what that looks like. So here I have kind of a cartoon example, but it's based in real life. It's based in some of Hopi Hoekstra's work from Harvard with mice. So we have in this sort of cartoon example, on the left, I have population 1 with the blue box, and that mouse has a dark coat color. And you can imagine that that dark coat color probably helps that mouse survive better in environments where the substrate or the background is darker. So a forest, for example, could be where you might find that mouse. Whereas there might be the same species of mice or same species of mouse in a slightly different habitat. So here I have population 2 in red on the right. And that one might have a lighter coat color, and that might be because that population lives at the beach. So if I actually took those mice, if I took a bunch of brown mice, and I took a bunch of light colored mice, and I put them in both habitats, and I asked how high is their fitness, how well do they survive and reproduce, I would expect to see what we see in this figure. In a forest environment, I would expect for the dark colored mouse to have higher fitness. But in a beach environment, I would expect the light colored mouse to have higher fitness. There's no universal best. There are trade-offs across environments. And that's what we call local adaptation. This is a really important process. It helps to maintain variation across populations. So if different areas favor different varieties, then for the species as a whole, we have variation. I'm showing here some stickleback fish that might be familiar. So these are the same species of fish, and we see different variations maintained depending on if you find those fish in a marine environment or in a lake environment. In some cases, this can lead to speciation. Hopefully you remember from your evolutionary biology that speciation is a process that takes a long time. And so adapting to different environments can be the start of that process. And if reproduction is halted, then those populations could become different enough that they're different species, but it's not a guarantee. And for organisms that can't move, like plants, local adaptation is one of those things that scientists talk a lot about as a way for organisms to cope with climate change. They might just need to adapt in real time to the climate as it's changing. And that may or may not actually be successful, but we hope it will be. So because this is such an important process, I sort of started thinking really early on, and whenever I was interviewing for grad school, what I was talking about was understanding the variation that underlies this process, identifying variation that enables organisms to adapt to lots of different climates. So we can revisit this picture that I showed earlier, and I just added a couple details and I changed the definition at the top. Before I was just talking about the process of adaptation, and here I'm just adding a genetic component to that. So this word antagonistic pleiotropy, it has multiple meanings. The way that I'm using it in this context is alternate alleles are favored in distinct habitats. So we have a fitness trade-off, and we can also think about how a certain allele could underlie that trade-off. Perhaps the reason that the mice in the forest have higher fitness is because they have the A allele. And perhaps the reason that the mice at the beach have a higher fitness is because they have the B allele. And my research was about identifying this. When I was an undergraduate, I was really into plants. I was studying plants, I was taking plant classes, and I took an economic botany class. And that's when I met this person. So this is Dr. Kenneth Olson. And I was already sort of having these ideas and these interests. And I was required, as you are right now, to go to a seminar for my class. And he was giving the seminar, and it was super cool. And he talked about identifying genetic differences during domestication and in different environments. And that was why I applied to Washington University. It was really that one seminar. I also applied to lots of other schools and I interviewed with lots of people, but I had already met this person. I met them during their talk. I emailed them after. I interviewed with them. We had a great interview. And then I went and worked with them for 7 years. So it's really just like that. You just talk to people, just keep going to seminars. The Olson lab generally studies plant adaptation, and we all study this from a genetic basis, but there were lots of different systems. So I studied white clover, but different people were studying rice, weeds that grow in rice fields, a little crop called Camelina sativa that we use for oils, and so forth. So my PhD research, my dissertation title was cyanogenesis, which I'll talk about in a second, and the genetics of local adaptation in white clover. Picking a research project is like really hard, I think. It took me, I got into grad school, it took me over a year to just decide what I was going to work on. But it came down to sort of three things. Whenever I was trying to figure out what I wanted to study, I first was driven by my personal interest. So hopefully by now you kind of know what I like to talk about: plants, adaptation, genetics, something like that. I also focused a lot on support. Whenever you're doing a new research project, we know different amounts of information about different species and different study systems. So part of why I chose white clover was because it was something my advisor knew a lot about and had worked on for many years. So there was sort of an ease of getting into that work. There was already some support in the lab. There was already sort of some infrastructure. We had protocols for this species, and our lab tech had been working on it for a while. So I felt like I wasn't alone, which is important in research. And it was also this study system that I'm about to tell you much more about was really good for my career goals. So I realized during my second year of grad school, I realized that research might not be the thing I wanted to do for the rest of my life, that I didn't know if it was going to fuel me for my whole career, but I loved teaching and I just wanted to work more and more with students. And so something about the white clover system was that it allowed me to teach, to bring together topics from many different subjects and use that to mentor students in research. So this was, I did a lot of work and some big projects, and I had a lot of help from my advisor and my lab and the greenhouse facilities and my PhD committee. But really, I had a lot of help from students. And I had a lot of help from students at multiple universities. So, so something that was wonderful for me during my PhD was just being able to be sort of a central organizing figure for a lot of people, that I was able to train a lot of students while also collecting a lot of data for my own research. So I wanted to go ahead and acknowledge all of those people here at the beginning and just say that everything I'm about to tell you, I did not do alone. Okay, so going back to this idea of antagonistic pleiotropy, this idea that different alleles could be favored in different environments. And I wanted to study this in white clover. Some of you may already know about this system. If you took ecology within the last year or so, intro to ecology, we've integrated this into our class. So you're gonna learn more about it today. This is a perennial legume. That means that perennial means it lives for many years, and legume means it has a special symbiosis with rhizobium bacteria in its roots, and those fix nitrogen. So because of that, this plant was used as a natural fertilizer for many years before the Haber-Bosch process was developed to make synthetic fertilizers. The way it reproduces is using the little white flowers that you see. It is an obligate outcrosser, so it has to have something help it. It can't have sex with itself. It needs a bee or something else to come and pollinate it. But it does also grow laterally along the ground. with these stolons. And so I'm showing a picture here of the stolon, and you can see at each node, there's little roots coming out. And so that enables it to grow and get big and sort of cover the ground. And so if you're familiar with it outside, it's sort of it intermingles with grasses. But from a scientific perspective, that's also really helpful because I can take chunks of this plant and I can put them in different areas and it will keep growing, and I have effectively genetically cloned it. So I like to say I cloned lots of plants, and it sounds really fancy. And really what I did was I cut plants into pieces and then I put them in different places. But that counts too. This plant is native to Europe, but it since has become widespread into many climates. In particular, the Dutch spread it around on purpose for agricultural purposes. And it was introduced to North America about 500 years ago. It's still used as a major temperate forage crop. And so we use it a lot to graze cattle and sheep. There's a lot of people that study it in New Zealand as well. Part of why the lab that I worked in was studying this plant was because it had this cool trait called cyanogenesis. So this is something you learned about in ecology probably. This is a trait that is the production of hydrogen cyanide upon tissue damage. So it's only when the plant's actually getting eaten that this is going to be activated. It's a trait that's fixed in over 3,000 plant species, so it probably evolves relatively easily. One thing that makes it unique in clover is that it's polymorphic. So not all plants are able to produce cyanide, only some can. And so it's a trait that varies in the environment, that varies in populations, and that varies across space. To be cyanogenic, you need two biochemical components. You need cyanogenic glucosides, which is a sugar ring with like a cyanide group sticking off of it. They just make those as part of their metabolism. And then you need linamorase, which is an enzyme that will chop off the cyanide group, and that then releases what we call the cyanide bomb. So these two components are located in different parts of the cell. So for those of you who are maybe taking cell biology, one of them is in the vacuole and the other is in the apoplast, which is sort of the extracellular area between cells. And that is how, because of that spatial separation, that compartmentalization, that's how this doesn't get activated all the time. It actually requires cell damage for the poison to be released. Okay, we know about the genetics of this. It's really just two genes. I'm oversimplifying. One of them is like a 3-gene cluster that is physically linked in the genome, but we'll call it 2 genes. So to make the glucosides, you need the AC gene. To make the enzyme, you need the LI gene. And in the evolution of this trait, both of these genes have been deleted multiple times. And so really the presence or absence of the 2 biochemical components is very simply controlled by the presence or the absence of these 2 genes. If you have both genes, we give you capital letters like this, and you are toxic, you're cyanogenic. But if you're missing one or both of the genes, then you are not. So what I'm showing down here are the possible cyanotypes. And the way you want to think about it for this talk is really the plants could be toxic or not. This trait has been studied for over 60 years. So people have known about this trait. They've known that clover had it. They've known that there were differences within clover populations. And people would go out and they'd measure a bunch of clover plants and they would calculate the percentage that had the trait. And they did that for over 60 years. They looked across latitudes on multiple continents. This has been, the same pattern has been found on multiple continents. They've also looked elevationally up the side of a mountain, for example. And more recently, they've even studied this from an urban to rural gradient. And so that urbanization gradient. And what we find is that generally in warmer climates, we have a high frequency of this trait. So if we go to Louisiana, somewhere in the South, Florida, we'll see a high frequency of cyanogenic plants. If we go further north, across space, we see this clinal pattern where there's a gradual decrease in the frequency of this trait. And so it goes from being 85 to 90% common in the South down to about 15% in the North. I'm not going to talk that much about why. It's still kind of an open question why this pattern evolves. A lot of research has tried to figure it out, and there are good hypotheses. I have some down there. But for the purposes of this talk, the point is that it does evolve, and it does so over and over. This trait doesn't become fixed. There are places where it seems to be good and places where it seems to be bad. This is also, whenever we see this general pattern that we call a cline, where we see a gradual change in a trait over space, this is also more evidence for local adaptation. So I was interested in local adaptation, and this was a plant that was already showing evidence of adaptation for a particular trait. And so with that knowledge, I wanted to know more generally what else could be contributing in this species. So far, we've looked at one trait and 2 genes, but this is a whole organism, and I have a feeling that just the chemical defense is not the only thing that matters. So I wanted to take a genome-wide perspective and use newer technologies to study everything else that could be contributing to local adaptation. I also wanted to know how important cyanogenesis actually is. We see these patterns that have evolved over and over, so can I actually detect that in my plots? The way that we tend to study local adaptation in the field is by using reciprocal common garden experiments. So I have sort of a graphic here showing what that is. Here we have 2 different populations of plants. One population has white flowers, one population has dark flowers. We plant both of them in 2 environments. So let's say the white one originated in the top of the mountain and the dark one originated at the bottom. We're going to plant both populations at both sites. And then we're going to compare the relative fitness at each site, just like that mouse example. So again, I'm looking in multiple environments and I'm asking, do I see fitness trade-offs? We can make this a little more genetic-y if we use genetic mapping populations. So we can actually figure out what's the underlying genetic basis of this adaptation if we use plants that are genetically related to one another in our experiments. Oh, maybe. Okay. So that was sort of my goal. I wanted to get some plants and clone them and plant them in multiple areas. And I wanted to use genetic mapping populations so that I could measure their fitness and determine what the underlying genetic differences were between them. I needed a large sample size. So I needed to generate about 6,000 plants. And I also needed to do this in multiple places across the United States. So when we proposed this, we proposed doing this in Duluth, Minnesota, in St. Louis, Missouri, and in Florida. And we proposed 2 different comparisons. So I will make 2 different mapping populations, and I'll put each of them in 2 different spots. So you'll see these words, this DG, that's my Duluth by Gainesville, Florida population. And SG is my St. Louis by Gainesville population. I'll come back to how I made those. Before I did this though, you can imagine I was telling people what I wanted to do and they were like, can you do that? Can you actually make that many plants? Can you actually measure what you want to measure outside? And can you detect differences reliably that justify doing more of this research? The reality is that research costs a lot of money. And before you ever even do it, you have to plan it and you have to show people that it's possible. So I did what I like to call my not so small pilot experiment, where I took 15 populations that I had collected from across the United States. I cloned them 3 times. I took 10 from each of these populations. I cloned each one of those 3 times. I planted all of them in St. Louis, and I ended up with about 500 plants. And I just kind of went with it. You know, I had a plan, but I also didn't know what was going to happen. So I put all these plants in the ground and then I took care of them for a while. I wanted to measure their fitness. I wanted to be able to compare them. So one measure of fitness was just growth. And I had a student who was excited about this, and we took all these pictures of plants that were growing along the ground, and we found this cool software, and we figured out how we could quantify that in a fast way, because we needed to be able to compare hundreds of plants really quickly. And so we used this thing called easy leaf area, and we could quantify growth. I also had this student, Dan, who now has his own PhD, and he helped me a lot with this experiment. We also counted every single flower that these plants produced. I think they produced over 55,000, which is not very many compared to my later experiments. These are the main takeaways from that paper. If you're interested in reading it, it is published in the Journal of Heredity. But essentially what we found was we saw evidence for local adaptation. So my two graphs over here on the left are both related to growth. So they're measuring growth. These two on the right are both related to floral production. They both measured how many flowers they produced. The top two are comparing growth and reproduction to distance from St. Louis. All of the plants were grown in St. Louis, but they came from places that were all over the country. So the general pattern was that if the population was closer to St. Louis, it had a higher fitness in St. Louis, which isn't that surprising. They were locally adapted. The further you got away, the population did worse in St. Louis. That was true if I just looked at relative distance, just how far the populations were geographically. But I also figured out how to compare climate. So I downloaded a bunch of climate data, and I actually asked, how climatically similar are all of these populations to St. Louis? And so if I look at growth and reproduction and compare that to climatic distance, I also found that populations that were climatically more similar had higher fitness in that climate. And the more different they were in their climate, the lower their fitness was. So generally just evidence for adaptation, evidence for differences between population, and showing that I can measure growth and I can measure floral production. And both of these seem like traits that are useful to study. Okay, so again, just revisiting my questions, I really want to get at the genetic basis though. I still haven't done that. And I want to be able to understand how much this cyanogenesis trait matters. So I picked my 3 locations. We have collaborators in Duluth and in Gainesville and in St. Louis. And I need to actually make these mapping populations. So to make a genetic mapping population, you kind of just do what you all did in like intro to genetics. You take 2 individuals that are different from one another, and usually you want a fixed difference. So here I'm showing they're homozygous for different alleles. You cross them and you make an F1 population, and that F1 population is half of one parent and half of the other, and they're all pretty much identical. But when you get to the F2 population, so if you take a bunch of these F1s and you cross them with each other, that's when recombination starts to happen. And what we end up with are a whole bunch of F2 plants that have recombinant genotypes. And we can think about that as being like a mosaic of the 2 parental genomes that we started with. We can then look at different regions of the genome. So here I have this green line, I'm just saying like at the top of the genome. And we can ask, what alleles do the organisms have? Do they have parent 1 alleles or parent 2 alleles? These have parent 1 alleles. These have parent 2 alleles. And then I can ask, are those 2 different genotypes correlated with a fitness trait that I measured for that whole population? Here I'm showing an example of a non-significant. There's no difference between the AA's and the A homozygotes and the B homozygotes. There's no difference in the fitness trait that I measured. But maybe at a different part in the genome, there is a difference. Perhaps at that region of the genome, being an A homozygote actually gives you higher fitness than being a B homozygote. So this is the idea of what we're doing. We're taking these F2 genetic mapping populations and we're actually putting those in the field, because that will allow us to later look at correlations between fitness and genotypes. Okay, so numbers, I needed to make 500 different genotypes for each mapping population. I need to replicate those 3 times and then put them in 2 different sites. And that got me to 6,000 plants. I'll very quickly show you how I did that. This took 2 years. So I started with 3 plants. 1 came from Duluth, 1 came from Florida, and 1 came from St. Louis. And all of the genetics we're studying originated in those 3 plants. By hand, I rubbed flower parts together and I made a bunch of seeds and I planted those in the greenhouse, and that gave me 100 F1s in each of my populations. I then realized that making 500 F2s by hand would be terrible. So I went to Lowe's and I went to fabric stores and I Googled and I figured out that you could buy solitary bees online, and I built cages in the greenhouse and I released solitary bees, and they very happily pollinated all my plants and I generated a ton of seed. I then planted all that seed. That gave me 1,000 F2s, but I needed them to be replicated because your experiments need replication. So I cloned all of those 3 times and in multiple environments. And so here you can see what the greenhouse looked like at various stages. And I will add that as an undergraduate, people who are helping me with this research, there was labor involved. There was labeling, there was working in a greenhouse with dirt, there was squatting outside and counting flowers. And so research can look very flashy, but can also be somewhat monotonous. And I kind of like that too. I had to actually get all of these plants to different locations. And so I had this whole system. Every plant had its own baggie. These plants can actually be wrapped up in like wet paper towels and transported, and they'll live for a while. So I literally put these plants in bags, and I put those in bags, and then I put those in boxes, and then I drove them 10 hours to Duluth or 14 hours to Gainesville, and we put them in the ground. And then I trained people what to do once they were there. And as you can imagine, oh wait, did I already, oh no, I missed my, there, okay, yes. As you can imagine, I was very tired. So here I am in Florida after I installed my 3rd and final garden. My husband was quite a rock during this whole process for me. And then once we got them there, we measured fitness for 2 years because ultimately we wanted to correlate fitness with genotypes. These are all the things we measured. Cyanotype doesn't change. So for every plant, we just knew what their cyanotypes were. Couldn't tell by looking at them, but we had that data in a spreadsheet. We also in the field measured growth and survival. So if you see green in the next few slides, that's growth. If you see orange, that's survival lifespan. And then we measured reproductive output. So if you see purple and pink, you're looking at how long plants flowered or how many flowers they made. So just looking at different aspects of fitness. And we measured all of these things for all 6,000 plants for 2 years. Oh, okay. Once we had all the data, I had a couple sub questions. So the first one was, do we see local adaptation? And boy, at this point, I hoped we did. Luckily, we did. So I already kind of accidentally showed the results, but because I planted the exact same genotypes in 2 different environments, I could actually compare fitness in one environment to fitness in the other environment. And if I get this negative correlation, that's evidence of a trade-off. That means that plants that were doing the best in one environment were actually the worst in the other environment, and vice versa. And that's what we saw. Obviously, this is messy data. These are big numbers, and the things we're measuring are hard to measure well, but the patterns don't lie. And what we saw over and over in both populations, so in the DG population at the top, and somebody's unmuted, and in the SG population at the bottom, we saw these negative correlations that showed us trade-offs across environments. Plants that did particularly well in Florida did not do well in Duluth and St. Louis, and vice versa. And we saw that pattern both for growth and for reproductive output. So good. There seem to be genetic differences that contribute to adaptation. Then I wanted to ask, is cyanotype related to that at all? You would expect to see, I have the 4 different cyanotypes in different colors here. And if certain types were better in certain environments, you'd expect to see some clustering. So I've tried to show that here with the figure where I've got kind of the blue on the top of one and kind of the black and gray in the bottom. But the reality was we didn't see that at all. We found basically no evidence that this trait that had been studied for over 60 years had any effect on fitness. Okay. So then I'm like, great, they seem to be adapted, but cyanogenesis seems to do nothing. So what is the genetic architecture then? What are the portions of the genome that are contributing, and how are they doing that? I had to, for this analysis, I had to actually create genetic markers for all of my plants. I used what is called genotyping by sequencing. So this is it's a type of sequencing where you get portions of the genome sequenced with big gaps in between. It's cheaper than whole genome sequencing. And for organisms where we don't have a really good reference genome, it's easier. We use enzymes basically to chop up the genome into lots of pieces. So I have to extract DNA from all of my plants. And then I have to individually chop up all their genomes. And then there's a whole procedure in the lab where we do what's called the library preparation. And then we send it off to a company and they do sequencing. And essentially the way that the sequencing works is for all of those chunks of the genome, you get like a little bit of sequence on the end, and then they send you back a data file that is bigger than you can open in Excel. And so I could go into more detail with this if you want to talk more about it, but I don't want to be too overloaded with genetic sequencing technologies. But I guess what I will say is at the basis of these technologies are restriction enzymes and PCR. So if you know what a restriction enzyme is and you know what PCR is, then you have the ability to understand pretty much all of the sequencing that we do. It's just complicated versions of that. So you get back all of this data, and then you have to be a computer scientist because you have to use Linux, you have to use code, and I have a friend that is very good at that who helped me with some of that. But now in the days of like big data in biology where we can get genome-wide markers for thousands of plants like I did for just this one project, you need to be able to do some coding and some computer science. So after a lot of work on a computer, and I should also say about 8 months of lab work, so a lot of lab work and a lot of computer work, we got down to about 2,500 SNPs. SNP stands for single nucleotide polymorphism, and that is basically just a marker in the genome. So we have a genome now with lots of markers, and this will allow us to correlate genetic markers with traits that we measured for these plants in the field. When you look at this genetic linkage map, you can think of each of these vertical lines as chromosomes. So we're kind of looking at the genome here. You can think of the horizontal lines as markers. So each one of those is a place where those original two plants that I crossed differed genetically. So we have traits or phenotypes that I measured in the gardens. I have SNPs, which are genetic markers. And I'm going to take those two sets of data and I'm going to correlate them with one another to identify QTLs, or quantitative trait loci. A QTL is a region of the genome, and it can include many genes. So this analysis that I did, just because of normal limitations, just technological limitations, doesn't really get me down to a specific gene, but it will point to regions in the genome that are of interest. Okay, so essentially, for every single one of these markers, I'm going to go through the genome and I'm going to ask, is it correlated with my fitness trait or not? And we're literally going to do that for many traits for every single marker. Whenever we find one that is significant, when we find a QTL or a region of the genome where the markers seem to be correlating with fitness, then we put a bar next to it. So I'm going to show you some results in a second, and it's going to be a map like this, a linkage map with all these chromosomes, and it's going to have a whole bunch of these bars. And I will walk you through what to focus on. But I just want to say, when you see a bar, it means those markers are correlated with fitness. Something in that part of the genome is helping plants adapt to different environments. I wanted to know how many of these are there. I had no clue. Where are they? If I measure reproduction and growth, are those controlled by different parts of the genome, or do they co-localize? Are they actually controlled by the same part of the genome? And for the areas that I identify, which one of the alleles is actually the better one? So if I find a region that's associated with reproduction, is it my Florida parent or my other parent that is increasing the floral count? Okay, results. There's a lot of buildup to get to results. So this is my Duluth, Florida cross. This is my more extreme cross. I've marked on this map the 2 cyanogenesis genes. So if cyanogenesis matters for fitness, we're going to see some bars pop up next to those. Again, we have color coding. So green and orange, we're thinking growth survival. Purple, pink, we're talking about flowers. These are the results. So you're looking at where various colored bars are. Different colors correspond to different traits. And very few, if any, get anywhere near the cyanogenesis genes. We really identified about 6 hotspots. So in the whole genome, there seemed to be about 6 areas where these continuous quantitative growth and reproductive traits seem to be affected. And when I look at the other cross, so remember I had 2 different mapping populations and 2 different comparisons, I saw the same thing. I didn't see any QTLs popping up near my cyanogenesis genes. And again, I saw a handful, in this case about 5, major hotspots where multiple traits seem to be mapping to the same location in the genome. This was particularly true for chromosomes 10 and 15. And so I have those circled here, and you can see there's just a whole lot of little colored bars right next to those. So then I wanted to ask, for those areas, do we see antagonistic pleiotropy? Can I actually see a trade-off between the alleles in different environments? I had predicted that I would see this for cyanogenesis, but that was not supported. So I definitely did not see a trade-off related to cyanogenesis. But I did see trade-offs related to growth and reproduction. So I'm zoomed in here on chromosome 10 or linkage group 10. And at this particular spot in the genome, we had both growth, so vegetative area, and reproductive traits mapping to the same location. And they did that in multiple environments. And so there's 4 bars here, which means that 4 different traits I measured all correlated with this region. One of those traits was vegetative growth in Duluth. If you were a plant living in Duluth and you had a Duluth allele, you grew more. For flowering duration, and I should say you actually were 86.7 centimeters larger. For flowering duration at that same point in the genome, if you had a Florida allele and you were growing in Florida, then you flowered for 20 days longer. If you had a Duluth allele and you were growing in Duluth, then you made 20 more flowers. And if you had a Florida allele and you were growing in Florida, you made 40 more flowers. All of this together shows that the alleles from that parent's environment increased fitness in that environment. Having the home alleles gave you higher fitness there.
Okay. Really quick, Sarah. We have about 5 minutes before we get to some questions.
Sounds great. That's good. Okay, and I should say I have these sort of asterisks. All of these were significant interactions too. So there's a way to test for whether that they're significantly crossing, and they were. What I saw interestingly, and this is I love this slide, but I also have to kind of walk you through it. What I saw that was really cool was that no matter where I looked, and so like which environment I was looking in, plants that had alleles from the southern parent favored growth, and plants that had alleles from the northern parents favored reproduction. And so you can see I have a dotted line in the middle here. On the left of that dotted line, I have vegetative area. And if you look at these sort of go up. Obviously, there's a lot of variation in the traits, but it looks kind of like a positive correlation here, and then again here, here, and here. And what that means is that having the parental allele, I hope I said this right a second ago, having the parental allele from the north led to more growth. We see the opposite on the right of this dotted line, where these correlations are going down. And that is because having the allele from the south led to more flowering. So I have sort of at the top, what is the takeaway here? We actually have antagonistic or opposite allelic effects at these hotspots in the genome that reflect different life history strategies. If genetically you have northern genes, you're going to prefer to grow over flowering. But if genetically you have southern genes, you're going to prefer flowering over growth. Okay. And what we actually saw, I think I'm going to skip this slide for time, but I'm going to go to my conclusions and it'll come back a little bit. So we saw clear evidence of adaptation. We saw that there were trade-offs across reciprocal environments. We found no evidence that cyanogenesis contributes to that at all, which was surprising for us. We thought it would. We saw that local adaptation could be characterized by a pretty small number of hotspots, QTL hotspots in the genome, 5 or 6 areas, and that those showed allelic trade-offs. And when we looked more specifically at the direction of those allelic effects, we saw divergent life history strategies. Specifically, we saw early investment in flowering in the south versus delayed flowering and long-term persistence in the north. And that appears to be what allows these plants to adapt to the different climates. It's really about trade-offs in growth versus reproduction. And this seems kind of like a paradox because we already know that we have all that data supporting cyanogenesis. And so it's still kind of an open question of how do those cyanogenesis clines evolve and when. I think it's happening at the earliest life stages. So I have this sort of model for how adaptation works in this species in my head and how that relates to climate and cyanogenesis. When plants are very, very, very small, that is when they're the most vulnerable to being eaten. That is when I think selection on cyanogenesis establishes these clines. So it's sort of that early life stage is when selection is most important for cyanogenesis. But I didn't actually plant seeds in my gardens. Remember, I grew them in a greenhouse and then I cloned them and I had those chunks of plants that I put out in the field. So I kind of missed this whole stage. Once the plants are already big, I would guess that herbivory doesn't matter as much. If you lose one leaf, it's not going to kill you. Plants in the north are dealing more with colder temperatures, and so they need to grow more to be able to persist through from one season to the next. Plants in the south had extreme mortality in my experiment. Most of them died before they even made it to the second year. And so really they need to flower so that they've reproduced before they die. And plants in sort of the middle kind of experience both. They grow more, but they also have to deal with harsher, warm conditions in the summer. Something's happening. Sorry, my slides are freaking out. Oh, I went too far. Okay. Okay, and what we saw, and this is, sorry, that kind of like screwed up my flow, but that's okay. What we also saw, I'm almost done too. What we also saw was that for those northern populations where they didn't flower as much because they were growing more, that paid off for them, because in the second year, if they survived the winter, they flowered quite a lot. So we have these huge peaks, which are representing floral reproduction in the second year. And those are all from the northern populations, because down here at the bottom, all those dots along the zero, those are all my dead plants in the south. So basically, flowering in the south is important in year one because you're going to die. But in the north, it's better to grow more, get through that winter, because then in year two, it's really going to pay off. Okay. Cool. So that's my journey, my research. I love teaching, and I did a lot of extra stuff in grad school. So I want to pause and take some questions, but I also have pictures, and I'll hang around if people can to share extra stuff. And this is one of my students. Thank you for listening. I haven't given a research talk in a little bit.
No, that was excellent. Thank you so much, Dr. Wright. We have so many questions. I know we're not even going to get close to scratching the surface, but there are a number of questions. Some of them you answered already, but there were some specific to your methods that you used that I think I had as well. So one of them, your pollination method was really quite interesting. How did you confirm that pollination was a combination of the two parents and not from individuals in the same population?
So when I did hand crosses, I was completely controlling who was pollinating who. And whenever I put them in the bee cages, I was only putting F1 plants in there, and I wanted them to be random. So I was like, I don't really care who's pollinating who, as long as it's all happening in this one group. So I had one cage for my DG mapping population, and then I had a separate cage for my SG mapping population. I think, does that answer that?
I think absolutely.
Okay. Yeah. There's a lot of organization in my project.
Did you lose any bees?
And yes.
Find them in unsatisfactory ways.
I see what you're saying. So that's a funny story, I guess. For the bees were sacrificial. So there were times when I was like talking to people at Lowe's, and I was like, I'm building bee cages for my research, and they were like, oh great, you're gonna save the bees, and I was kind of like, actually not. But the bees that I purchased them from a company that like grows them and gives them to farmers to like pollinate their crops, or like in their backyards. So if you go to crownbees.com, you can read about this company in Washington. They'll ship bees around. They come in little cocoons. You can hatch them out, and then they're solitary, so they don't have like hives or anything. So I got them as little cocoons. I hatched them. They lived only within my cage, and then they passed away in the cage. Some of them did reproduce and leave little cocoons in. So I tried to like give them the environment that they could reproduce, and some of them did. Did they ever like escape? So there was one time that I was coming back from a field site with my undergrad, and I was like, let's check the greenhouse real quick, because I just started a bee cage. I just want to make sure that everything's cool. And as soon as we walked in, I saw a bee that was on top of the outside of the cage that flew away. And then I was like, oh my gosh, this is the worst thing that could happen, because I can't have pollination going on in my greenhouse where like there's not supposed to be any. So we spent that afternoon cutting off every single flower we could find in the whole greenhouse, and we did that for about a week just to make sure that there wasn't like extra seed being made outside. But that only happened once, and it was like, yeah, it was a slight panic, but I think we got it under control. Anyway, the fun stuff that happens in research that you don't expect.
Exactly. So while I'm on that, someone had asked your favorite and least favorite field research experience.
Oh boy.
You may have just told us the least favorite, but
oh gosh. So okay, I don't know if I can come up with like a single experience, but generally, I just love the day-to-day of being in the field and watching the field change over the season, and seeing the different like seeing a bunch of like aphids come, and then a bunch of ladybugs come, and then the ladybugs all lay their eggs, and then there's ladybug larvae, and then there's butterflies, and then there were groundhogs, and then just sort of this like seasonal transition of all of the insects, and just kind of being in that and part of that for multiple years. It's hard to quantify what you can gain just by like observing and being immersed in an ecological system, even though it's kind of a controlled one. So I loved that. And then I loved working with students. I didn't talk about my student projects, but there were like 10 different student projects as well. Least favorite was the fact that I had very little control once my experiments started. So I could control the breeding and I could control the labeling and all this stuff. But once they were in the field, if there was a flood, I just had to deal with it. If they were flowering a lot one week, I just had to work more. If, it was just kind of like dealing with whatever was going to happen. Like there was a huge rainstorm one year and so forth. So that could get discouraging, but I had to be very persistent. There were a couple tearful phone calls to my advisor, but he kept me going. So yeah.
Perfect. Going right out off of that question, how did you make sure the collaborators in those other locations in the other states took all the same measurements and precautions and state of non-control, like you said? How did you make sure that you were still?
Yeah. So there is something to be said for big numbers. You can get away with a lot of noise if you have big numbers. And so a lot of my critiques were, can you do more though? And I was always like, no, this is as big as I can go. Like reviewers of grants, but how about larger sample sizes? So that, big numbers help, but I did have to train them. So I had, for my pilot experiment, I had protocols that I had developed. And so when I went to the sites, we had already recruited students. The students were pre-recruited so that when I arrived, like we installed the gardens together. And then I was in constant contact with team leaders at each site. Essentially, it was counting flowers and removing them. So that was pretty easy. And then taking pictures at regular time points. And then I would analyze all the pictures. So there was a lot of quality control with the later analysis. But yeah, I will admit that there were, I worked on this for years and it was a lot of work. And there were times when I was like, but what if I find nothing? And so I was honestly shocked and pleasantly surprised that I did find the really strong patterns that I found. And that I did find these really cool allelic trade-offs that were very consistent. It was like Florida alleles want to flower, not grow, and Duluth and St. Louis alleles want to grow, not flower. And I saw that over and over. I presented like a condensed form of my results, but anyone who's done research knows that you do 30, 50, 100 times the amount of data analysis just to get to those final, like, here's the main takeaway. So I saw that pattern over and over, and I was honestly shocked that despite all of the ways that things could have gone wrong, the signal still sort of came through. So anyway, I hope that answers that question. Yeah.
This is my question. Were you able to identify any specific genes in those linkages that you pointed out at the end there?
Not yet. That would have been great. So if the like, what would have given me like a science or a nature paper, I think, which is for students, that's like the coveted best journal to publish in or whatever, would have been finding the cyanogenesis genes like overlapping with QTLs. And then I could have argued maybe it's those genes that are actually driving, but I didn't find that at all. So, but because of the genetic resolution I had, there are lots of genes within those regions. Even in those hotspots where I'm like, oh, these traits are mapping to the same spot, I don't actually know if it's the exact same gene or if it's just a couple of linked genes that are just like close together in the chromosome that act in different directions, possibly. So research goes on, and once I left the lab, there's a grad student there who is making F3s and who is doing more genetic mapping. So we can get more details by continuing to cross the plants and doing more studies. And now that we have these candidate regions, we can really focus in on those areas for like more intense sequencing and stuff like that to get at like, well, what are the possible genes in those areas? So it'll take more to figure it out specifically. But I think a lot of students, especially with plants, I think there's this perception, and not even students, just the public. There's this perception that like we can take any gene that we want and we can just pop it into a plant with genetic engineering and we can get the phenotype we want. But a lot of times the phenotypes we want are quantitative. It's more seeds or faster growth. And those are controlled by lots of genes. And often we don't know specifically which genes they are. And we really only know that for like maize, like corn and rice. Most plants, we don't even have that ability. So disease resistance genes are often another one we can figure out and that we can cross in or edit in. But we still have a long ways to go to actually understand like what genes do what. So anyway.
Another research question. What gene was analyzed in the white clover genome to see the correlation between the allele and the fitness in the F2 generation?
Okay, so I was always looking at a particular marker. So I did all that fancy sequencing to generate a whole bunch of markers. And it's actually like those markers that I'm using to correlate with fitness. So I don't know what gene those came from, if I'm honest. I just know that that's a position in the genome where the two parents were different. And so there's a whole lot of genetic analysis that happens where it's like, I'm using markers as proxies for regions in the genome. And then once we sort of identify a general region, then we can get more detailed as to like what genes are in that region. But I don't actually know the specific genes. I just know which markers I was using. And in the computational analysis, they all get names that are very long and crazy. And it wouldn't be helpful to tell you what it was if I knew it.
A more general question, is white clover considered invasive in North America?
That's a good question.
If not, why not? Like, when do we stop calling something invasive when it's so entrenched?
Yeah, that's a good question. So is it invasive? I don't know if I actually know the answer to that. I would say no, but it's certainly doing very well. Often it's referred to as a human commensal, meaning so a commensal is like a species that benefits without harming or helping the other species. So like white clover really benefits from us. It likes to be where we are. And that part of that is because we like mow the fields and stuff. And so because it has this growth habit where it grows along the ground, it can't get very tall. It can't compete with like if you mow the grass, it can compete with grass, but if you let grass grow or you let something turn into a prairie or a bunch of wildflowers, it will often get outcompeted. So I wouldn't call it invasive sort of because of that reason. Like it can't just like totally take over. It kind of needs us a little bit, I think. But it's certainly prolific and it spreads really well and it makes lots of seeds, but it also makes lots of flowers, which means we get lots of honey. So I would argue that even if it were technically invasive, humans have already altered all the habitats anyway, and this plant really supports bees, which are in decline. So I would argue it's still good, but I'm partial. And that was just an on-the-spot response. So I don't. Maybe someone else is like would define that, but I don't want to play with invasive definitions.
I'm going to finish with a more personal question. Throughout your career, did you ever question what you were doing?
Yes, so many times. I talked about counseling at the beginning, right? Yes, yeah. I was always like, do other people care about this? Is this cool? Is this a good use of my time? Am I going to be able to teach one day? I want to teach college students. Am I actually going to be able to do that? In the 8 months of lab work, I was like, am I going to get like, so I did all this lab work after 2 years of field work. And I was like, am I going to get genetic data? What if I can't get all my genetic data? And like, there were failures along the way before successes. So yes. And talking to people helps. Something about research something that I felt in grad school that I don't know if that's common everywhere is that because everyone has a very specific project, you can feel very isolated. You can feel like no one else, like I was kind of the expert in my area and no one else was really doing the exact same thing, although there were some people doing similar things. So it can feel very isolating, and it's really important to like force yourself to keep interacting with other people and collaborating and going to conferences. And I know I can be more introverted sometimes, and so I had to like work harder to sort of talk to my friends about their research too, so that they would ask me about mine, so that I wouldn't feel alone in some ways, if that makes sense.
Exactly. I asked that question because I knew the answer.
Yeah.
It's the same for all of us, which I think is good for students to see.
Yeah. Oh, and
it's good. It's really important, and we want to thank you so much for taking the time to talk to us. Oh,
what I also just want to say, constant imposter syndrome. If you think everything I did was awesome and looked great, then I didn't fool you, but like I convinced you. But like before this talk, I was so nervous. So there is sort of this feeling of like, yeah, just always, I think if you're a good scientist, you're always questioning yourself and you're always questioning your work, because that's important. But it also means you're always like, is this the right thing though? And at some point you just have to say, I read the papers, I know this plant, I'm going to argue for this, and I'm going to stand by it. And if someone else disagrees with me, like, that's okay. So yeah, anyway, okay.
Excellent. Thank you so much, Dr. Wright, for joining us. Thank you to everyone who's here and the questions that you sent in. I'm sorry we couldn't answer them all, but please reach out to us. I know Dr. Wright would be happy to talk to anyone who wants to go see her at some point.
Yeah, email me. I have a website. Google me. If you Google me, you'll find my website. Happy to chat more.
Perfect. Thank you so much. And we hope to see everyone next time with Kristen Blevy has on April 8th at 12:30. So we'll see you then.
Awesome. Thanks, everyone.